We're covering concepts 3.5 and 3.6 in this presentation. That's going to include proteins and nucleic acids. Proteins are quite important in living systems. They consist of more than 50% of dry mass of both cells, and there's all kinds of functions including defense, storage, transport, enzyme function, etc. This diagram, the next two slides show the various protein functions. So enzymes are critically important. They're made up of proteins, and they act as catalysts to speed up chemical reactions. The building blocks for proteins are amino acids, and these are organic molecules with a carboxyl in an amino group. So amino and carboxyl. The central atom is the alpha carbon, and the other group that's important is the side chain, and there's going to be various different chemistries that can be in that side chain R group. And the 20 amino acids known as the building blocks for proteins are different by their differing side chains, and we see the groupings here based on the chemistry of the side chains. This group, the side chains are hydrophobic, like anywhere from a simple hydrogen with glycine to linear and branched hydrocarbon structures like leucine's a branch structure. There's also ring structures like phenylalanine and so on. These are all hydrophobic. Polar side chains are included in this group, and these are going to be interactive water, so these are going to be hydrophilic and polar. Another set of hydrophilic groups will be your charged group. So you have amino acids side chains that are going to be negatively charged at pH 7. You're going to have amine containing side chains that are going to be positively charged at pH 7. So these are all going to be hydrophilic. So as you start forming polypeptide chains and proteins, you link your amino acids together by peptide bonds. And a polypeptide is just a polymer of amino acids, and they can range in length from a few to 1,000 monomers. Each polypeptide has a unique linear sequence of amino acids with a carboxyl terminus and an N terminus. So you got one end of this is a 3 amino acid chain, and it has a amino terminus, N terminus, and it has a C terminus, carboxyl. Peptide bonds are formed by dehydration reaction, so you lose the water to form the new peptide bond. And the polypeptide has a repetitive backbone, purple, in which side chains extend. So functional proteins consist of one or more polypeptides folded and twisted in some unique three-dimensional shape. And So this example shows two different ways to show that shape. This is a lysozyme that we have present in our sweat and tears and saliva. Its job is to remove bacteria by digesting them. And you can see the difference between the ribbon model in A and the space filling model. So your sequence of amino acids determined by the gene encoding the protein leads to the protein's three-dimensional structure, and this structure determines its function. Like in this case, the antibody has a three-dimensional structure that will precisely fit with a specific flu virus protein. So, backing up here, there are four levels of protein structure: primary, secondary, and tertiary, and quaternary. So the primary structure is the unique sequence of amino acids. Secondary structure you'll find in most proteins. There's kind of two types. There's an alpha helical coil, alpha helix, and a folded beta pleated sheet. Tertiary structure is the overall three-dimensional space and structure the protein fills. And part of what stabilizes tertiary structure are the R groups, and the R groups can interact and form bonds, such as hydrogen bonding between polar side groups. You can have hydrophobic interactions, van der Waals interactions, ionic interactions. Two cysteines can actually form a disulfide bridge or covalent bond. Quaternary structure, that's going to involve more than one polypeptide unit, more than one protein. Here's an example of quaternary structure, transthyretin, that consists of two separate protein subunits. The next example is collagen. It has three separate protein subunits that are braided together in this helical coil structure, but each color represents a different protein. And hemoglobin has four protein structure subunits, four subunits. It has two alpha subunits and two beta subunits. And a single amino acid change in the structure of hemoglobin will totally change its function to where it severely limits the way the hemoglobin can carry oxygen, and it limits the flexibility of the red blood cell. This is sickle cell disease. So just a single amino acid change, this number 6 valine, totally changes the way the hemoglobin works, the way the blood cell is shaped. So in addition to primary structure, physical and chemical conditions can affect structure. If you alternate pH, salt concentration, temperature, and other environmental factors, you can cause a protein to unravel, which is known as denaturation. And this loss of the protein structure, this denaturation process will eliminate biological activity. The biological activity is lost in the denatured protein. So it's hard to predict a protein structure from its primary structure. So most proteins, they go through several intermediate steps of folding until their final stable shape is determined. Scientists use X-ray crystallography to determine 3D protein structure based on diffractions of an X-ray beam by atoms of the crystallized protein molecule. An example of this is Roger Kornberg's Nobel Prize-winning work, where he used X-ray crystallography to build a 3D model of the protein RNA polymerase II. As you can see here, this gray structure, kind of a Pac-Man looking structure around the DNA, which is blue. And it determined potential functions for different regions of the protein, including how DNA and RNA are held in place for the transcription process. So DNA is blue, and here red is the RNA structure. And this is all developed from their model based on X-ray crystallography data. Moving on to nucleic acids, the thing we need to know about that is that proteins are built from nucleic acids. DNA is transcribed to RNA, which then is translated into proteins. This is a central dogma of biology. And the genes are made of DNA. That's a nucleic acid made of monomers called nucleotides. There's two types of nucleic acids, DNA and RNA. And DNA provides the blueprint for its own replication, as well as a blueprint to synthesize messenger RNA, which then it gets translated into protein. So nucleic acids are polymers called polynucleotides. Each polynucleotide is made of monomers called nucleotides. So this square here is your nucleotide. And each nucleotide has a nitrogenous base, a pentose sugar, and a phosphate group. Excuse me. Each base has one or two rings that include nitrogen atoms. So in DNA you have thymine, cytosine. RNA you have uracil instead of thymine. And then both DNA and RNA you have adenine and guanine. The sugar in DNA is deoxyribose. And ribose has the extra OH group. So deoxy is missing that OH group that you see in ribose. The sugars carbons are numbered. In fact, they're given a prime number, 1 prime, 2 prime, etc. when you build them into a nucleotide. So adjacent nucleotides are joined by covalent bonds that form between the OH group carbon, so the 3' carbon, with the phosphate group on the 5' carbon. So here's the 5' carbon, here's the phosphate group. So you're joining adjacent phosphate and hydroxyl groups. And this makes up the sugar phosphate backbone of the DNA or RNA molecule. So RNA molecules usually exist as single polypeptide chains. DNA molecules have 2 polynucleotides spiraling around an imaginary axis, forming a double helix. And they run, the 2 strands of DNA run anti-parallel, so one runs 5' to 3', the other one goes from 3' to 5'. And this is referred to as anti-parallel. The nitrogenous bases pair up to form hydrogen bonds. Adenine always pairs up with thymine, and cytosine always pairs up with guanine. So the linear sequences of nucleotides in DNA are passed from parents to offspring. So this is your genetic information, or inherited information. And 2 closely related species are more similar in DNA than more distantly related species. But you can use molecular biology to see how close evolutionarily you're related to another species. So there's a couple of concept summary slides here that wraps up all the key concepts in this chapter. So now you have completed chapter 3.